I think we'll get started. It's my great pleasure to introduce Julia today as our seminar speaker. This is a seminar co-sponsored by Genetics and the Morgridge Institute. And I'm so glad we were able to convince Julia to come all the way from Scotland to visit us. And so Julia, she's Argentinian and Italian, but she grew up in Argentina. She went to college there. And after her college, she did her PhD with Ross Kagan at WashU. And after that, she decided she was tired of the US, right? And went to the UK, where she did her postdoc with
Owen Sansom.
Yes, yeah. Owen Sansom. And and that was at the Beatson Institute for Cancer Research. And after that, she stayed on in the UK, and she became a junior, then senior research fellow at a different cancer research institute, Wolfson Wold Cancer Research Institute at the University of Glasgow. And she has been, since 2016, she has been a group leader at the Beatson Institute for Cancer Research, also in Glasgow. And since 2022, she's a professor and Wellcome Trust Senior Research Fellow, which is a very fancy thing to be in Europe, and at the University of Glasgow. And her group has been doing some beautiful work studying intestinal regeneration and tumorigenesis using the fly model. And she's looked also at intra-organ communication, and more recently at how the tracheal system crosstalks with the intestinal stem cells. And I think this is going to be part of her talk today. And I'll let her give her talk because it will be much more entertaining than my introduction.
No.
Welcome.
Oh, I really would like to thank Daniela for this very, very kind invitation. I always, it's always a logistical nightmare for me to organize a trip, international trip. And so when I got her invitation, I really could not say no. It was really a great pleasure to meet you last year virtually, and I'm very happy to meet you in 3D now. So, and thank you all for sticking around to come for the seminar in spite of the bad weather. I know many people had to go home earlier, so I really appreciate those of you that have stick around, the ones that people have connected in Zoom as well. So thank you all. And I'm hoping to be able to give you like a sort of overview of the research that we're doing in the lab, and I'm happy to answer questions afterwards. So as Daniela said, so my lab is interested in studying the intestine, and need to make sure that I can move this. Okay. So we study the adult intestine, and as you know, as one of see it as this organ that is a central regulator of many processes that go on in our body. And it's really a very, like, has many physiological functions, and it's a major barrier epithelium, but it also has key immune and metabolic roles. It's the biggest endocrine organ in our body, and also through the gut-brain axis controls many higher order functions as well. So we are interested in studying how the health in this organ is achieved and what happens when it goes wrong at the gut level, but also more systemically. So to answer these questions, we use flies. See, this is okay. So we use flies as a primary model system, the adult fly midgut, which is a very close analog to the mammalian small intestine. And I mean, this is a cartoon of it. You will see more real pictures of it. But really what this reflects is that there is a clear anatomical difference between these tissues. However, the cellular composition, the physiological roles of this system, the fly system, and also the molecular mechanisms that regulate fly gut health and disease are very much conserved to those in mammals. And And of course, we have the beauty of Drosophila genetics that allows us to really individually visualize, but also genetically manipulate in time restricted manner different cell types in the fly gut. And this is a marker, for example, that will label the intestinal stem cells, key players in the gut, in gut biology. And it's undifferentiated progeny, which is called enteroblast, which is the cell type that is going to give rise to the different specialized cell types in the gut intestine, but it's also an important component of the stem cell niche. This will become more important later in my talk. I just wanted to mention it here. So we have genetic tools that allow us to visualize these cells, but also to manipulate them. And also we have the enterocytes, which is one of the specialized cells in the system. They are high absorptive and metabolic roles, and you can see them here in a real picture. And we have an enteroendocrine system in the fly gut, which is very much like has many important sensing and secretory functions, and it's really an important player in the communication of the gut with the outside world as well. So what is key, quality of the fly gut, if you wish, is that as the mammalian system, it has a very strong regenerative capacity, which is due to the presence of the stem cells. So you can challenge the gut of the flies to regenerate, and we often do that by feeding the flies with chemical agents or with pathogenic bacteria. So we feed the flies with these agents. This will cause damage of the intestinal lining, mainly the enterocytes. And this, damage of enterocytes produce signals that will then also lead to the proliferation of the stem cells, which is essential for the recovery or the repairing of the gut epithelium, but also to keep the health of the whole organism. If you prevent stem cells from this regenerative capacity following damage to the gut, soon after the animal will die. So in real life, this is how this process looks like. This is a wild type, like a control or homeostatic gut, and this is how the gut responds after damage, acute damage for maybe overnight or a couple of days, depending on the agents that we use. See a very strong expansion of the stem progenitor compartment, very high proliferation that we can actually quantify by staining with, for example, phosphohistone, an antibody against phosphohistone, which will label mitotic cells, which are very much the only quality of the stem cells in the fly gut. So this is a direct measure of stem cell proliferation. And you will see a lot more of these type of graphs during my talk. So you can also induce pathogenic proliferation in the gut by genetic means. And you can do that, for example, by knocking down oncogenes, by knocking down tumor suppressors like the APC, adenomatous polyposis coli, which is the main driver of colorectal cancer in humans, or you can do it by overexpression of oncogenes like activated form of RAS or SARC, activating chronic inflammation by overexpressing ampairs. Flies ampairs are the interleukins or inflammatory cytokines that activate JAK-STAT signaling. And so we can also do recapitulate pathogenic conditions in the fly gut. And that looks very much similar to what happens when we use, cancer in mammalian models. There is an expansion of the CRIP domain, which is the domain that contains proliferative cells. So these are qualities that make this system very interesting and very useful to study regeneration and also tumorigenesis. So keeping this in mind, so the overarching interest in our lab is to really understand how changes to gut homeostasis caused by the damage, inflammation, or tumors trigger local responses of the gut, that is, stem cell intrinsic adaptations or interactions between the gut and the microenvironment that are triggered in response to this type of perturbations. but also, you know, how this type of adaptations of the gut have an impact at the whole body level, which then also is not a one-way road. This will inevitably feed back to the gut. So we do use damage and regeneration in response to damage just as a major paradigm for our studies, because we know also that a lot of the processes that are acutely activated to resolve damage in the gut can be chronically activated in tumorigenesis. So often we study the gut in the context of regeneration, and then we see how much of that can also be reflected in or playing a role in tumorigenesis. And so when I talk about damage in my talk, it's going to be mainly a pathogenic damage. We use by feeding flies with pathogenic bacteria, and I will tell you otherwise if it's a different type of damage. So, and I will start with, you know, some of the work that we've done on stem cell intrinsic and gut niche interactions in the intestine. And one focus of our lab in the last few years has been to understand the interaction of the gut with a particular component of a very prominent component of its microenvironment, which is the vasculature. So these are blood vessels in the mouse intestine, and this is the tracheal system that is akin to the vasculature in flies and also to the respiratory system. Trachea there is no blood that goes through the trachea because flies have an open circulatory system, but trachea, like blood vessels, are in charge of delivering oxygen to the target tissues. And so in both cases, as you can appreciate, this is really a very prominent tissue associated to the gut epithelium, but there was at the time very little known about whether there was an active role of this vascular system in maintaining homeostasis or regeneration of the gut, and how was that working. And so the work that I'm going to show you was mainly driven by postdoc Jessica Poreshon in the lab, and also a large part of the work that I'm going to show today was done by Jade Phillips, a PhD student has recently joined the lab. This is an electron microscopy of the gut epithelium, and pseudocolor here is intestinal stem cell. This is an enterocyte. There's a visceral muscle layer that sort of covers the gut tube, and in between these you can observe terminal tracheal cells, which they are similar to the tip cells of vasculature, and that they are really the cells in this vascular system that are in direct contact with the target tissues, and in this case with the gut epithelium. So you can see that there are these projections of tracheal cells which have very prominent cellular structures that go in between the epithelium and the visceral muscle. And in the same is the case here. So they are a very intimate component of this gut microenvironment. So we wanted to know whether there was a functional role of this system in gut regeneration. And so the first observation in this project was that we saw that when we induce local damage to the gut epithelium, there was a very striking morphological change in the trachea associated to the gut. And that is very much a non-cell autonomous response to the damage is not reaching the trachea, it's within the gut. So this immediately told us that there is some sort of communication between these tissues. And so you can appreciate here is that this is a single tracheal body with many cellular projections, and these are stem cells that are always around the areas where these branches extend. And this phenomenology is conserved in the mouse intestine, where we can see an increase in CD31 positive endothelial cells around the areas of regenerative crypts following damage. We also see that fly guts bearing tumors or hyperplastic due to the action of oncogenes are also highly vascularized when compared to a normal gut. And this is also the case in the mouse tumor models. We have done, this is an RNA scope to label PCAM, that is a marker of vascular endothelial cells, and you can see that there are more PCAM positive cells surrounding tumor areas than normally in the normal tissue. So that's all good. We have like a sort of phenomenology that is conserved, and then we wanted to see whether there was functionality for this, gut tracheal vascular remodeling in response to changes to the gut. So the first experiment that we did was a very crude one, but we thought this is some sort of like a deal breaker if it doesn't work. So we've used genetics to completely ablate tracheal cells in a time-restricted manner, and just before inducing regeneration of the gut, and we wanted to see what does this change or affects regeneration. So this is how it looks like. We use a tracheal specific driver for terminal tracheal cell specific driver to drive a pro-apoptotic gene, and that's very efficient to ablate most tracheal cells, not all, but most of them. And we see that these guts that have ablation of the trachea are much less effective as regenerating than a normal one. This is how proliferation of stem cells in control conditions following damage, and this is in the context where trachea have been ablated. So really, trachea have an important function. They are necessary for the gut to regenerate. And also, if we put animals in hypoxia chambers and we induce damage to the gut, there is also a significant to a lower extent, but significant impairment of regeneration following different types of damaging agents. The same thing is for the trachea. This is by no means a pathogenic damage specific thing. It happens with whichever agent we use to damage the gut. So there is a significant drop in the regenerative capacity to the gut when the animals are in hypoxic conditions. So trachea and also oxygen are necessary for intestinal regeneration and tumor growth. But so this is us. This is a summary of years of work, but it's published, and I don't like to spend a lot of time with this. But after we did a lot of work on this project to find out the mechanisms of this crosstalk, and what we identify is that the production of reactive oxygen species that is a first response to pathogenic and other damages or other perturbations of the gut. These ROS are an important signal that is sensed by the trachea, and this in response to this, there is a molecular program that is activating these tracheal cells, which include the activation of hypoxia-inducible factor, FGF receptor signaling activity. There is also an activation of neuronal genes, which was unexpected. These trachea look like neurons, but they are not neurons. But in the context of regeneration, where they are really become so plastic and so changed in their shape, they do activate neuronal genes. And they knock down trachea specification factors that are normally required in the embryonic stage for these tracheal cells to be specified from progenitors, which was fascinating because it means this plasticity comes with some sort of naivety or less specified characteristics of these cells. So all of these things we tested functionally, and we know that are required for the gut, for the trachea to remodel in response to damage, and this remodeling is necessary to provide oxygen, effective oxygen to the tissue, but it also comes with the production of some angiokine factors that are made by the tracheal cells, like FGF, which activates FGF receptors, signaling in intestinal stem cells, and that's necessary to drive proliferation. Yeah. Now, there were some observations that we made during the course of this work, which suggested that there was more. Obviously, we always thought that would be the case, but that there's more than ROS playing a role in the system. And one thing that we saw that was kind of puzzling at the time was that, so, ROS, the production of ROS is a way of, killing pathogens. And it also would cause, these pathogens would cause, or damaging agents would cause cell death in the gut. But what we observe is that if we use genetic means to block the cell death of enterocytes, the reactive oxygen species and stress signals that are activated in this tissue as a response of this ROS are still all there. However, simply blocking genetically cell death of enterocytes was very effective at impairing the remodeling of the trachea, and it also impaired proliferation of the stem cells. But also, we know that remodeling of trachea happens in conditions or changes in the gut that do not involve the production of ROS. For example, when we induce damage with DSS in the gut, there is, increased tracheogenesis, increased proliferation of stem cells. And if we use antioxidants in this context, that really does not impact either the trachea remodeling or proliferation of the stem cells, whereas in the context of pathogens, the NAC will make a difference. So the question following up from this was that, there are hypothesized there are gut signals other than or in addition to reactive oxygen species, which are playing a role in the remodeling, inducing trachea remodeling during regeneration. And so one thing that is very obvious when you look at the midgut during the process of regeneration and recovery after damage is that it undergoes very significant morphological changes. And that is mainly reflected by guts becoming shorter and thicker. And this is a reversible process once you remove the damaging agent and regeneration has happened after the peak of stem cell proliferation, there is a restoration of cell shape. And the remodeling of this gut-associated trachea is also a dynamic process and a reversible process. You can, in white here, there is an individual tracheal cell that has been traced throughout this process. And as you can see, after regeneration, during the recovery phase, this tracheal cell, has lost a lot of these very prominent cellular branches and return to its original shape. But what was very interesting is that even when we look at guts that are regenerating and we look regionally, we observe that this is a region that has been geographically defined by other researchers and named as R4. This is the region of the gut that undergoes the most prominent changes in cell shape during regeneration. And in this region, there is very high levels of tracheal remodeling during damage. Whereas if we look at R5, which is a much less, an area of much less cellular changes, the tracheal remodeling is less prominent than in R4. And this was very interesting. And where we were taking these observations in the fly gut, I stumbled across a paper which I found fascinating, which describes the adaptive vascularization that happens during pregnancy in mouse. This was a mouse paper, and they show that parts of the belly that are, that undergo the biggest changes, the skin in the belly that undergoes the biggest changes during pregnancy, have areas of very active vascularization and also groups of stem cells that are proliferating to induce the skin remodeling. So we thought, well, this could be something going on here, and an association of morphology of the gut with the adaptive tracheogenesis. So that would suggest that there is an actual physical, perhaps, interaction between the cells in the gut and trachea. And this is something that we wanted to test, and we have used a system called GRASP in flies that was developed by neurobiologists to really look at synaptic interactions and between neuronal cells. And basically, we have a reconstitution of GFP in the context of two cells, when two cells are in very close contact with each other. So what we did was to use a promoter of the trachea fused with one half of this GFP and an enterocyte promoter with the other half. And if there is physical interaction between two cells, we should see GFP signal. So the trachea would always constitutively have a RFP on it. And indeed, we saw that when we induce damage and regeneration, then we start to see the expected remodeling and increasing tracheal tissue, but also we start to see these points of interaction with the intestinal epithelium. So suggesting that indeed this is quantified here, there are dynamic contacts between the cells in the gut and the trachea. We've also used live imaging to visualize these morphological changes in the gut. This is in a control condition. And these are guts that are undergoing regeneration. And you can possibly see, I don't know, it's not so clear in this image, but you can see that there is a stretching in and out of epithelial cells. We can actually do, how do you go out of this? Okay. We can really segment these different stages of the epithelium, and we can see the outlines of individual cells. And what was quite obvious is that these cells become like longer and in the dorsoventral axis and shorter in the anterior posterior axis when the gut is regenerating, and then during recovery, they restore their shape. We have used tools to basically quantify this type of changes, and eccentricity is actually a measure of the loss of roundness in the cells, and the more the lighter yellow indicates areas of higher eccentricity, and this is just quantified here. And there is more eccentricity during regeneration, and that's restored or like reverted during recovery. P is the damage agent that we use. And we using this, we can also quantify the dorsoventral axis and anterior posterior axis length, which is reduced in the context of damage and restored afterwards. So these are all indications that cells are undergoing clear morphological changes in the gut and could be released signals to the trachea. Now, in all the contexts that I have shown you, there is damage involved. So there are many factors that are going on. So we wanted to know whether there are ways that we can induce, like change the morphology of the gut without inducing damage. And so for that, what we did was using we fed the flies with methyl cellulose, which is a substance they will not be able to absorb in the gut. So they basically inflates their belly as they eat it. And also it causes something very similar to what happens when we use damage. The guts become shorter and a bit thicker, but there is no actual damage proliferation going on. So it causes the same changes in eccentricity that we've seen, or similar to what we've seen during damage, when there is more eccentricity and there is a decrease in anterior posterior axis, and also these type of perturbations, like this use feeding on cellulose in the cell, in the guts, is sufficient to drive the remodeling of the trachea very actively. So we really believe that there is mechanical changes in the gut that are important drivers of this the tracheal remodeling. And how is this translated? How this translates into changes in tracheal remodeling? And for that, I mean, we did long story short, but we looked into mechanosensory channel piezo because it's known. I mean, it's a channel that is activated by pressure, by mechanical cues, and it has been shown to work in the vasculature and be activated in the vasculature by shear stress, and it regulates vascular development in different contexts. So we looked into when this could be also playing a role in the flies in the trachea, and we use a reporter of piezo, gene expression, and also protein reporter. And in all cases, we see that there is a massive activation of piezo expression at the protein and RNA level in the trachea in a reversible way, dynamic way, that matches with the gut regeneration and recovery. So we use genetic tools to knock down piezo from the terminal tracheal cells specifically, and two different RNA lines, and we see that if we knock down piezo from terminal tracheal cells, that prevents tracheogenesis in response to damage, and it also prevents regeneration of the gut. So piezo is also piezo channel signals to the activation, the opening of this calcium-gated channel, and it activates the calcium influx in the cells. So what we use for calcium sensors to see in the trachea, and this is how it looks like in homeostasis. Very little is happening. Trachea is in red and the calcium sensor is in green. Whereas if we look at the regenerating gut, we can see like very, very intense flashes of calcium signaling within the tracheal cell bodies that expand to the cellular projection. So we still don't know. We still need to show that this signaling is dependent on piezo, but I thought it's nice. We are excited because it's a very recent movie and it has taken us a while to set up this live imaging. So that's really nice. And finally, I mean, so we looked at, we have taken a look at piezo in the gut, in the mouse, and we see that, and this is p-prime for the vasculature. Piezo is really very actively upregulated in the mouse intestine, not only in the vasculature, it's also very prominently activated in the epithelium. So we are doing experiments with knocking down piezo from the vasculature in mice and see what happens to regeneration, and let's see what we see. I'm not sure, and also in the context of tumors, where we also see an upregulation. So I'd like to finish this part of my talk just by summarizing what I've told you. So ROS is an important signal for tracheal remodeling, but there are also mechanical signals that are emerge from the gut during the process of regeneration or tumorigenesis. Most likely we still need to see that piezo and activates piezo in the trachea. This is important for remodeling, tracheal remodeling, and for regeneration of the gut. We don't really know yet what lies downstream of piezo and whether there are common signals to ROS. We suspect some might be common, but we are looking into that at the moment. Now, one other interesting observation that we saw from this work, and it has taken us to some additional work on stem cell intrinsic adaptations, is the fact that it's very clear that areas of high proliferation of stem cells require high provision of oxygen. In the context of regeneration, but also in the context of tumors, this is a RAS tumor, like a group of cells expressing activated RAS, and you can clearly see there is a lot of trachea there as well. So this suggests that oxygen is indeed required for stem cells to proliferate acutely or in more chronic processes, which is kind of counterintuitive with what the dogma for intestinal stem cells is at the moment, which suggests that oxidative phosphorylation which is a metabolic process that requires oxygen, is it really plays a role in stemness and differentiation of stem cells, whereas hyperproliferative conditions do use glycolysis as a metabolic route. So we decided to really, based on these observations, to really look into what this might tell us about metabolic adaptations and stem cells need to undergo during regeneration, and whether they are the same in the context of tumorigenesis. And this is work that is done in my lab by a PhD student and Tian Belek, a postdoc in Yashuan Yu, in collaboration with Oliver Maddox, also in our institution. And what we have done, this is very early work, much less developed than what I have shown you before. But basically, we decided to, so we feed flies with regular like sucrose solution or pathogenic bacteria, which is normally what we do for regeneration. And then we fact sort stem progenitor cells from these guts, and then we do RNA sequencing on metabolomics. And this is very early on, and just want to give you some of the examples from what we have found so far. We look at the RNA sequencing signature. We do clearly have a very prominent signature for mitochondrial constituents, like proteins and make like genes that encode mitochondrial structural proteins, but also components of the respiratory chain complexes, especially complex 1 and 4. Also TCA cycle components, that's OXPHOS, some amino acid enzymes, and some other more metabolic pathways enzymes. So this will be consistent with the fact that, mitochondria are actively being produced during the process of regeneration. Now, what is important to keep in mind is that this is a signature of not only the stem cells, but also the enteroblasts. And remember, I told you that enteroblasts are a component of the stem cell niche. So they do influence stem cell proliferation, but they are not the stem cell themselves. So we could have a mix of things going on here. So we have started to do genetics to functionally, assess these different candidates. And what we are doing is, use genetic tools to manipulate the gene expression individually in intestinal stem cells or enteroblasts, right? And we have started by looking, for example, this is just one example of, this is a core component of complex 1 that we know that if we knock it down in the stem cells or in enteroblasts, in both cases, it has a very strong effect on regeneration. So it's cell-autonomous free cells cannot proliferate without this in response to damage, but also in enteroblasts, it's also playing a role affecting stem cell proliferation. So possibly the function that this, and the same thing happens with this gene, which is a component of electron transport complex 4. In both cases, we see that both cell types need mitochondrial activity. We obviously need to understand how, but clearly, we think that the role of mitochondria in the different cell types could be distinct. And this will also help us to understand better what is the trachea and the oxygen provision doing. Is it affecting mainly enteroblasts, or is it playing a role in both, and how, and so on? So, and you might have noticed this is a very, one of the highest upregulated genes that we find. It is an enzyme of an amino acid metabolic pathway, and is required to convert glutamine to proline. And the reason why I wanted to point this to you is because it was very interesting. It's the only gene that actually, when we knock it down in the stem cells, it does nothing. So if we block expression of this gene in stem cells, there is no effect, the gut regenerates fine. But if we inhibit, knock it down in enteroblast, it has a very significant role in impairing regeneration. So that is actually something that is exclusively required within the stem cell niche. And we are very excited to pursue and understand better what is this amino acid pathway possibly doing here. Now, I told you that we have done metabolomics, and I'm going to show you what we have done. Is again, the metabolomics is much less developed because it has taken us a while to really be able to optimize the system to get a reliable signal. And we did a pilot experiment, and now we have done like a proper experiment with several biological replicates. So I'm showing you what it consistently comes through after our various experiments, which is a strong amino acid signature. So several amino acids have appeared to be highly upregulated within, again, stem and progenitor cells in the gut during regeneration. And there are two in particular that are very consistently always showing high. This is our pilot, and I will show you the final experiment with several biological replicates. Leucine and isoleucine, which are two essential amino acids, constantly come up highly upregulated. And the reason why we are interested in those two, apart from the fact that they seem to be very strong candidates, is that when we have done work on identifying transcripts that are changed in tracheal cells during regeneration, and we find mini-discs, which is a leucine importer, and is highly upregulated in the trachea during regeneration. So we are pursuing hypothesis that one of the ways that these tissues communicate is through the transfer of nutrients between the gut and the trachea, and that is also playing a role in this crosstalk between these tissues. So this is something we were starting to look at more based on the results of the metabolomics and the transcriptional profile that we have of the trachea. And if I have time, I don't know, do I have time? How much time do I have? Doc? Okay, I'm going to try to take less. I just want to take you a little bit back to my original slide where I show you the gut as part of a multi-organ system and as a central player of coordinating different organs, and the importance of understanding, gut health and how this might impact other systems, which is really the second main aim of our lab. And one of the really the observation that drove us into this research direction was this one. We was looking at different perturbations in the gut, again, damage, DNA damage, but also pathogenic damage or inflammation or tumorigenesis in flies, on similar perturbations in mouse models, or I'll show you more of them in humans. There was a very profound change in the enteroendocrine cells. These hormone-producing cells that are made in the gut, progeny of the stem cells that are very diverse cells, and they have the capacity to sense many. They make neuropeptides and neuroendocrine hormones that are also made by the brain, and they do have an incredible variety of interesting sensing capacities. They have other receptors, gustatory receptors. They just look like little neurons that live in our gut. The profile of these cells changes very dramatically during gut damage, inflammation, or in tumorigenesis in different ways. You might appreciate that tumorigenesis and this RAS activation in the gut leads to an almost complete ablation of these enteroendocrine cells, and then other contexts have more enteroendocrine cells than controls. Now, this is a very conserved phenomenology as well, what I'm showing you here is an example of a human colorectal cancer and stained with chromogranin A to look at endocrine cells, and they are known. And also mouse models, the same, but I'll show you more on that. So we were interested, I mean, our obvious hypothesis from this was, given what we know about the importance of enteroendocrine cells acting systemically, I mean, this type of changes must have systemic impacts, and what are they, and what is the functional role of these cells in this context? And really, we know from work that we did some years ago, but also recent work from many other laboratories, is that, you know, fly enteroendocrine cells very much have the very biological properties that are exactly like the ones in mammalians. And we, for example, discovered a subtype of enteroendocrine cells that have the capacity to regulate the gut proliferation locally, but they also can sense nutrients, in particular sugars. And in response to that, they can really modulate systemic functions, including energy homeostasis and feeding behaviors and so on. So the work in this project is being carried out by a PhD student, Andre Medina, also from Brazil. And Andre was, given these observations, it was interesting to study what the functional significance of this enteroendocrine cell dysfunction, disruption was in intestinal pathologies. When I'm going to focus on this phenomenology, which is the loss of enteroendocrine cells, which happens not in all type of conditions, but certainly when you have activation of RAS, which is a very common mutation in cancers of the gut, but other tissues. And there is a loss of enteroendocrine cells. There is the same similar type of phenomenology happening when we activate RAS in mouse. And also, when you look at human tumors, and this is an analysis of the DGCA data set, which includes 478 human tumors. And these are all different genes that markers of enteroendocrine cells, and you can perhaps appreciate all of them are significantly downregulated in this. So this loss of enteroendocrine cells is a very prominent feature of tumors that we know from mouse and flies. RAS activity is a major driver of this, and it's essentially an effect of high RAS activation in the differentiation of these cells. So what we did is, because of our knowledge, we know that these flies, these enteroendocrine cells can sense sugar, some of them. One thing we wonder is, okay, if these flies that have no enteroendocrine cells, what happens when you give them a choice of food, given that they have lost, we hypothesize they must have lost the capacity to sense the presence of sugars in the food. So what we did was some food choice assays in this animal. So we have animals that have wild-type guts or guts that have overexpression of RAS in their stem cells, and they have lost their enteroendocrine cells. So we give them a choice of yeast, or sucrose, and we can measure, quantify how much of each of these foods the animals eat. And what we see is that, you know, if when we measure the consumption of yeast food, the flies that have RAS activation have no special preference to yeast versus flies, control flies, or these are flies that have RAS activation, but we have also used genetic tools to restore enteroendocrine cells in these flies. But if we give them sucrose, when we measure the sucrose, we can differentiate which food they are eating. They do have a clear preference to eat high sugar food. See here, which is completely reverted when we restore enteroendocrine cells. So it looks like these enteroendocrine cells are really regulating the drive of these flies to eat food, to eat sugar food. And there is an obvious, and then we did another experiment, and we just, instead of giving them protein or sucrose, we did 2 different concentrations of foods with 2 different concentrations, one is very high, one another very low. And you can, so in both cases, they would eat more than than the control flies, but they eat much more of the high concentrated sugar than the low one. And then in both cases, you can restore this by restoring enteroendocrine cells. So somehow these flies can eat high sugars, you know, and in excess. And an obvious consequence of this is that they might be, for example, more prone to develop diabetes or other, you know, type of pathologies as a consequence of this. And this is something we are testing. But, you know, one of the things that we did was looking at these different conditions, the control. This is a control fly. Again, these are flies that have overexpression of SARC in the gut. This oncogene will drive proliferation of the stem cells. They will drive hyperplasia of the gut, but does not affect enteroendocrine cells and the RAS model. And we feed the flies with normal food or high sugar diet. And we measure insulin signaling systemically. And control animals or animals that have SARC overexpression in the gut activate insulin signaling by phosphorylated AKT. This signature, like it's a way to look at insulin signals. We look at total and phosphorylated AKT, and both of these proteins are upregulated systemically when we feed these flies with high sugar diet. But when we feed the RAS flies with a high sugar diet, there's no response, even maybe a little bit less, but not so sure because there's less. So basically, they do have complete loss of ability to respond to this high sugar diet. They have like sort of kind of insulin resistance. So we are very excited about these results because we know that there are ways that we can restore endocrine cells in the gut genetically, but also with in intestinal organoids. For example, they use MEK inhibitors to allow even normal organoids to produce a full complement of endocrine cells. So there are ways how you can induce the production of these cells. But what this tells us is that, you know, if you lose endocrine cells, you lose this capacity to sense, you know, the level of sugar that there is in the diet. And this, you know, can lead to leads to an increased appetite of sugar and systemic metabolic dysfunction. We still haven't done experiments where we restore endocrine cells, but we know that if you having proliferation of the gut is not sufficient, you need to have a loss of endocrine cells. Now, whether this is mediating signaling through the brain or directly on metabolic tissues, we have no idea. It's a very starting point, but we are very excited because we feel that there is a lot that we can learn from here. And I would like to finish by thanking my lab. I have mentioned all the people that have done the work that I presented today, so that's fine, our collaborators, and I would like to welcome Cancer Research UK for funding the research that we do. And thank you for your time.
So I was wondering about, you told us that cell death and keratinocytes was also repaired. So do you have thoughts on what the nature of that signal is? And I guess and kind of a related question is just the overgrowth that you see in the trachea in these areas where the stem cells are proliferating, is that driven by local hypoxia caused by the stem cells depleting oxygen, and then that's the signal? Or so anyway, those are two big.
Yes, yes, so. We have tried, we have not been able to demonstrate the presence of hypoxia in the gut during regeneration. We have like different reporters and sensors and so on. Now, this is what made us think about ROS, which we know is a is highly produced in regeneration. And it's also linked to oncogenesis, because there is evidence that the hypoxia-inducible factor can be activated by ROS even in normal conditions. So we don't think, we don't have any evidence to suggest that this hypoxia was diagnosed. Regarding the cell death, what we think is happening, and we have some evidence that I haven't shown. So when the lymphocytes die, they also are eliminated from the epithelium. And I think that in itself causes changes in the architecture and the organization of the tissue. Now, when we inhibit this death of the lymphocytes, we see this eccentricity that I showed you, that prominence during regeneration is prevented. So it does, we believe this is very directly playing to us tissue architecture and alleviating some of these mechanical changes, which you are forcing the remaining cells to try to occupy. That is an amazing system in the sense that when you remove an endocytes, some of the remaining ones try to compensate. They are and they try to compensate for the loss of that space, and until new ones are made. So there is really like the cellular dynamic is very fascinating. So we think this is what's happening when we increase the death. The signals are there, all the stress signals. We use the gases inhibitors. It's a very final thing what we inhibit. The rest is all happening. We still see all of the cytokines we made and RS, so we think it's more pain.
Beautiful work. So I wanted to follow up on the adaptive trichogenesis that you talked about. So clearly there's a lot of adaptation right after that. So what happens afterwards when you have a new equilibrium? Is that new trachea that's been put, does it return? Does it like undo, or is it just the new normal?
So what we see, the changes that we see in the blood are exclusively morphogenetic. We don't see changes in the number of fragments. So it seems that there are more, but the fragment numbers, but when we count the bodies, they are the same. It's more like they just generate a lot of these projections. We are doing like imaging, which is something we need to do to really understand what's happening in the regression phase. We know very little about what's happening in the regression phase. We are taking different matters. The cost of like driving the project is taking that sort of angle for her own research career, but she wants to understand molecularly what's happening during the recovery phase, but also cellularly, whether this is severing of these projections or they are just retracting. We don't know. But yeah, but it's very, very, very interesting. Because you think, I mean, what we know, for example, I showed you that if we feed the flies with this methylcellulose, is sufficient to drive trichogenesis, but it's not sufficient to drive proliferation. To drive that, activating tractor remodeling per se is not sufficient to drive the proliferation of cells. You need other things. Now, if you activate tractor remodeling, when you keep this remodeling going after damage has been induced, this recovery of the gut is delayed. So there is an active role as well in this retraction, in just slow, like, restoring homeostasis in the gut. So, yeah.
There's a very cool ending to the talk. I think there's a lot of cool applications. Like that question at the beginning was a lot of issues that develop tumors show the kind of phenotype of vascularization of the tumor. And I was curious how universal the regulatory process of that was. Like, do you think that, or do you have people found that like case of needed mechanical stress and ROS is helping that vascularization?
No, I mean, this is something we don't know. It's not been done before. This is something we want to do. So we are doing experiments. So the tumors are a little bit more complicated because they require more genetics. So we are a bit lagging behind with our tumor work. See that the regeneration is easier because you don't need genetics to induce damage. So we can use the genetics to manipulate the tissue. So we have generated dual driver systems where we can generate tumors in the gut and label and independently manipulate the trachea. That's taken us a lot of time, but we are there. So now we can really look into whether these processes are we have very well understood or somehow understood in regeneration, whether they are playing also going in the genesis. With mice, we are doing that as well. They are using a vascular specific treat, not kids from vasculature. We're looking at the regeneration, but we are also looking at this tumorigenesis in the intestine. But because with the mice, we have also the possibility to do like endoscopic transplantation of tumors in the cecum, so we don't need to use genetics to generate tumors. But
one comment first is that the protein succinate is the best substrate for mitochondria in plant muscle. So maybe that's correlated to what you're finding or glutamine transforming to protein and maybe the activation of the
okay, I talk more about okay, yeah.
Two questions, quick. First, did you try to give insulin to the tissues before assaying for restoration of AKT?
No,
To see the levels of insulin release.
we are measuring insulin. We are measuring insulin production in these animals in response to the high sugar diet. But all we have is what I showed you. We are really just starting with this we have done this question so many times already, and it's always to see that there is like the levels of insulin activity, insulin signaling activity we observe in this trans over expression twice. In normal food is comparable to the ones that we see in high sugar food. So they are completely starting super high levels. And they have a huge level, they have totally insensitive to any. Yeah, to have more high sugar. We are looking into this production.
So that's question. One is, have you tried other ROS, other two radical speeds or just one? And the same question for antioxidants. And the way you give it, like diet or inject.
So for antioxidants, we have done an experiment with feeding and zinc. But we have also done genetics. And this is why we know that it's ROS. So ROS is not primarily from the gut. There is intrinsic ROS by the tracheal cells. But what we know is that if you use genetics to block the production of ROS from the enterocytes, you prevent the tracheal remodeling. It's less effective than the systemic, but this is because the systemic is blocking ROS in other cases are also available. So that's for blocking ROS with our genetics and plasmids. The other question is if we have used other oxidative damage or
Yeah, not oxidative damage. So I guess you give superoxide or some.
No, so when we use damage, we have done DNA damage by bleomycin, which also causes ROS production, and that also drives.
Have you tried inject instead of eating?
No.
There are no further questions. What is the conclusion?